Okay, so far we have talked about DNA is from a chemical standpoint and a physical standpoint. So we know about the DNA structure, the double helix, and the basic model. We've also talked about DNA replication. So we now know that when we start out in G1, I take one of these pairs of part of the homologs, we take that and we divide it and we produce two chromatids from each homolog by the process of replication. What I wanted to do now is I wanted to talk a bit about what does DNA really look like and what does it look like certainly in G1. What are the major components, at least we're talking about in eukaryotic systems. And then I also want to talk about how do we get that piece of chromosome compressed down into these very tiny chromosomes that we see finally in late prophase, early metaphase when we're in either meiosis or mitosis. So just to kind of take a look, if we were to go in and look microscopically at chromosomes in normal G1 phase, as you see here, much of our DNA would look like this. We would have beads on a string, as it's called, and we have these little round structures, and then we have these interconnecting pieces that are DNA. And so the round structures contain DNA, but it's actually protein wrapped around by the DNA itself with an interconnector that goes in between. During G1 or during certainly the G0, which is when cells are differentiated, much of your DNA, much of your chromosomes, it actually looks like this. There is some areas that are more condensed, such as around centromeres and telomeres in some areas, but for the most part, much of your DNA actually looks like these beads on a string. Now, by the time we get to late prophase, we've gone through the replication process, and we now have these structures that look like this. Each of these are dyads, so that's a homolog of that particular one right there. It's arranged in what's called an ideogram. We'll talk more about these later in lectures, but this is the compressed form, the maximally compressed form. And in reality, it looks something like this. Here's your sister chromatid 1 on this side, which has a piece of old and new DNA due to replication. And then your sister to that on this side over here, connected by a centromere region that's in here. And they do look kind of fuzzy and outlined like this. So really the question is, how do we go from what we think of as chromosomes, particularly in G1, to this dyad configuration that we have here, this highly compressed form that we find in late prophase, early metaphase. Now, unfortunately, you look around the web, on books, this is what people draw a chromosome looking like, is this structure here. And they don't look like that, okay? Only once they're in this highly condensed form, do they look anything like maybe this structure over here. If you were to look at DNA in most of its life, it would look something like this. It would look like this double-stranded coil wrapped around these little beads, if we're thinking about it here. So unfortunately for you, you've learned, been taught that a chromosome looks something like that. Chromosome looks like this. In fact, if we were to go into a eukaryotic cell, into the nuclear envelope, we would see that this is really what it looks like during most of its time. It's very spread out. The DNA is all diffused throughout the entire nucleus region. Only after some massive compression, of course, after replication, and then through G2, do we actually end up with these things that look like this type of chromosome up here. Okay, these highly condensed forms are certainly like these things that you see in pictures where we've taken these chromosomes and actually taken an image of them, a laser image. That's a highly compressed form of our actual DNA going from here somehow to this. Now, put this in some perspective, okay? We think about fruit flies, they have 4 pairs of chromosomes, or 8. In humans, we have 23 pairs. 22 of those pairs are autosomal, so they're foreign, and yet 1 pair, which is the sex chromosomes, but they're still chromosomes with lots of different traits and loci found on them. Now, that gives us 46 total chromosomes, which accounts for about 6 billion base pairs, right? Now, if we go back to our Watson-Crick model, we think about, okay, about 0.34 nanometers per base pair, the distance between those. If we do just a very simple little bit of multiplication, take the number of bases that we have times the distances, we're looking at something about 2 meters, about 6.5 feet of DNA in every cell in your body, right? So every cell, every somatic cell at least, not the gametic cells, but every somatic cell has about 2 meters of DNA. Kind of give you some perspective on that. In this country, standard door heights are about and a half feet, 7 feet. So they are about 2 meters tall. I'm a fisherman. I like to fish. So many of our fishing rods, if you like to fish, are about 6 feet, 2 meters long, pretty standard size, unless it's a fly rod or something. And maybe for something some of you are more interested in, something like a wedding gown. Very often from the shoulders down through the train, that's about 2 meters on a standard, normal wedding gown that doesn't have a particularly long train, but a nice, beautiful standard train. It's about 2 meters. All right, now let's take that. At 2 meters, we've got just hundreds of thousands of millions of cells in your body. If you took all that DNA, we're able to stretch it out, okay, and pile it all together, it would literally reach mostly to the moon, almost completely to the moon from the Earth, just in your body. So just taking the amount of DNA that you have and stretching, linking it together, if we could do that with all the cells, would be this huge thing. And it really becomes a question: how do we get that into these cells, right? How does that 2 meters of DNA get packed into this nuclear region that's really fairly small, and it can be various sizes, but almost all of that 2 meters of DNA is found right there in the nuclear area. Okay. And then, of course, once it's even in there functional, how do you package it down then when you want to break the cell apart by either meiosis or mitosis and separate it? It has to compact even more. So kind of look at this in perspective here. We've got DNA, okay, and we know that DNA is double-stranded and helical. Now that itself is a compression, right? If you think about it, if you think about a spring or something, if you were to pull the ends of the spring out and stretch it, it would be quite long, right? But because it's coiled, it's already compressed. Now it's gotten wider because one thing of the string, spring is this wide, but if you coil it, it gets much wider. Well, helical DNA is already coiled. So we've already done partial compression without anything else going on. We've compacted somewhat. Okay. To do it any further, we're going to need some help. So we're going to bring in these proteins that are going to help us bind up that DNA into a series of finer and finer and greater and denser stages until we finally end up with what we think of as a metaphase chromosome. Now this drawing again is a bit fake, because if we had a piece of DNA like this, it wouldn't end up this big as a chromosome. It'd end up much smaller than even this little thing over here as a chromosome, because it's a compaction and it's going down further and further and further and further. So we're going to talk about how that occurs, right? Now for this particular lecture, I'm just going to break it into two parts. The first part here, I'm going to kind of concentrate on this area right here, okay? And then we're going to walk through the chromatin compression and condensation areas as we walk on the next ones. So let's think about the first one, right? Well, the first idea is we have to think about, okay, we know that there's protein and DNA associated, and we know that there's this compressions going on, this condensation process. What are some of the models? Well, there are two primary models that have been out for a while, for 40, 50 years. One was the folded fiber model, which is the idea that if you look at these chromosomes, they're individual units and they're packed out. Now, this fit a number of things. The width and things seem to be correct on these models, and there's some ideas. But what didn't fit and what made it not a very highly accepted model is this histone form the outer core idea. The basic idea here is that these specialized proteins called histones actually form a core of the chromosome, and that the DNA is somehow in the center of that core, like this, right? So you had this outside area, and the spherical DNA was in the middle, and that's clearly not correct. We know that that's not the way that chromatin or these beads on a string appear. So this has not been, but it did have some correct ideas about the width and other things. The reason I'm mentioning it to you is that folded fiber model pops up in Google searches and in books as the model. But in fact, it's not the primary existing model right now. The primary model is what's called the nucleosome model, and it's more widely accepted as the base model, and it's based on this structure called a nucleosome. Right? And we're going to talk a lot about what a nucleosome is in just a moment, but it's basically a group of histones, which are proteins, small proteins, and then the DNA complexed up together. Okay? And what we're going to try to do is talk about how we get from just double-stranded DNA into this nucleosome form that we have here. And then in the next lecture, we'll pick up on some of this other area, going from just single nucleosomes strung together to more tightly packed areas as we work our way eventually down to a metaphase chromosome. Okay? So let's think about just chromatin in general. Okay? Remember that we have to go back on that history lecture. We got to go on DNA introduction to DNA. That it was in the 1800s when chromatin was first identified, right? It was talked about there was this protein-like stuff and this other stuff, and it turned out that other stuff that was in there was DNA that made up chromatin. So Fleming in 1880 described what was going on. Now, what was there, it turns out, was double-stranded DNA, okay, helical DNA, plus these proteins. These proteins are specialized proteins called histones. We'll talk more about those in just a moment. We take a group of those histones and put them together and then add the DNA to it, and we get a nucleosome. And then this DNA connects to another nucleosome to form these structures that we saw in that first picture, which are the chromatin fiber, are known as beads on a string, right? So what we need to do is we need to look at this nucleosome and kind of get an idea of what the heck that thing is, right? Well, this is my cartoon of it. Okay, and again, it is a cartoon. These ball-like structures are the proteins, these kind of beach ball-looking things. We'll talk about this other protein down here, the specialized green proteins here. This yellow structure kind of appears supposed to represent my double-stranded DNA. It's basically wrapped almost twice around the structure called a nucleosome. So let's look at the nucleosome itself, right? Well, it turns out there are two big classes of histones, of these proteins that are involved. They're what are known as the core histones. That's these balls that you see here, kind of red and brownish balls that are here. There are 8 of those that make up a histone, but there are only 4 types, so there's 2 of each. There's 2 H4s, 2 H3s, etc. Okay? They're not particularly large by standards of proteins. They're about 120 amino acids in length, and they're complexed in and around. We're going to show you a computer representation of what this looks like more than my little cartoon in just a moment. But they're sort of found wrapped up in together as the center part. In fact, this area is often called the core particle. And that's good and bad because sometimes the DNA is called the core particle. It just gets confusing, but this is the core region of a histone. Okay, these histones are highly conserved. Okay, very important word. It'll show up on tests and things. Highly conserved during the evolutionary process means that they haven't changed much. The amino acid sequence that's there is very similar across species. And which means, of course, that since the amino acids are the same, then the DNA that originally produced and coded for that remains the same. Just to give you some idea, the H4 histones, if we compare humans, your H4 histones, with peas, Mendel's peas, so we're talking about the separation of plants and animals millions of years ago, right? The H4 histone only has about 3 to 4 differences between humans and many plants. So it's really highly conserved. It means that it's stayed constant throughout a long period of time, which must mean it's incredibly important and adapted for the purpose that it has. Okay. Also, we should think about these proteins themselves are very positively charged because they have a lot of lysine and arginine residues. If you're having biochemistry, you know what those are. If not, we'll talk about them later. But basically saying that there's a lot of the 120 amino acids are either arginine or lysine, which means they carry an overall positive charge. And we'll see that's important in a moment. We talk about how all this works together. The second class of histones are the H1 histones, represented by this little green part that we see off down here. Okay, these are a little larger. They're about 200 amino acids in length as far as making up this kind of part. They are not as highly conserved. In fact, there's even tissue difference in expression. So if you look at the H1s from, say, liver and maybe from neural cells, the H1 may have quite a bit of difference in the amino acid structure that makes them up. So they're not highly conserved, and they're certainly not highly conserved between different organisms. Okay, there's a lot of change that occurs, a lot of mutational differences between different organisms there. They are relatively loosely associated with the core particle, is what it's called, meaning that unlike these, which are tightly bound and sort of make up a physical structure, these actually connect only in a lighter way. Part of that is because they have multiple functions. One of their functions is, as you kind of illustrated here, is to hold all this together, to kind of pull it down and then stick it together. Okay, we're going to find out that H1s also have this affinity for each other, and that helps us later on when we start to pull these nucleosomes together to give us higher level structure. Is one of the ways that happens is by attraction of these H1 regions in some of their domains. So let's go back and look at this nucleosome here and just kind of review the parts. We've got this octamer, so 8 histones in the middle, okay? And again, each one of these is doubled, so they're there. It turns out that the areas of these histones, these proteins, the areas that are kind of on the outside of the nucleosome are hydrophilic. They're water loving, right? And the parts of them that are found toward the inside are more hydrophobic. So there is a sort of a chemical relationship here to the cellular water and to the areas of the types of residues that occur on these particular proteins. This yellow thing, as I said originally, is the double-stranded DNA. It's approximately 150 base pairs, around sometimes 170. With the linker between one nucleosome and the next, we say it's about 200 base pairs total. So each nucleosome is accommodating in total about 200 base pairs. And that's important, for problems and things that we all sometimes have problems where you see them. I've got a gene here and it's gene's 8,000 base pairs long. How many coils of the DNA, in other words, how many times would the DNA itself go around? So 10 bases, base pairs divided there. How many nucleosomes would I expect to find? Well, I'll just divide 8,000 by 200. Or if it's 4,002 divided by 200, whatever the base pair number is, 200 will do fine for government work, as we say. Okay. Then we have this H1 histone here that's going to play multiple roles, but right now it's helping to hold this whole thing together. And basically it's going to just sort of keep it in place, like a putty or something at the bottom that's going to work to help hold it together. We then have our linker DNA, which is a standard double-stranded helical DNA that just happens to connect to the next nucleosome, right? And then this whole structure then is about 11 nanometers tall. So it's called 11 nanometer fiber. Sometimes sometimes you see it as a 10 nanometer fiber, but I think the more recent idea is this is consistently about 11 nanometers from the top of this structure to the bottom of it. So we call it 11 nanometer fiber. This is important because when we start talking about condensation, we'll start talking about 30 nanometer fibers and 700 nanometer fibers and all that. But this is the base unit. It's about 10 nanometers, 11 nanometers across. Okay. And then the next thing we have, is that, as I said, there's a lot of these positive residues that are found in the nucleosomes. And what happens there is that because DNA is an acid, right, deoxyribonucleic acid, it has net negative charge due to those phosphate groups that basically are on the outside. You have a lot of positive charges found in these nucleosome proteins, these histones, and so you get electrostatic bonding that can occur. So that's one of the ways that we actually hold the DNA into this nucleosome is by this simple positive-negative charge relationship between negative DNA and positive residues that are found on the nucleosome themselves. So that's why I said it was important that these nucleosomes have these net positive charge. Okay. This is what a computer thinks this little model actually looked like a bit more. This outside surface right here is the DNA, the double-stranded coil going around, and then these inner blue and red and green and yellow are the histones that are found in the middle, right? And they're found in these areas. And this is maybe a side view of kind of what it looks like. So again, you've got your hydrophobic parts of these in here. You've got your hydrophilic ones more on the outside. Now, something that's very important we're going to talk about in a moment is you'll notice that there are these strings coming out right here. There's one right here. Those are called tails, and they're parts of the proteins themselves, the histones, and they stick out past the DNA to the outer surface. And there's several of them. There's strings that look like this. They're part of the actual internal histone proteins, but they stick out, and they're called tails. They're important for this reason. Okay, compaction of these chromosomes and this chromatin and all of these nucleosomes occurs because of a series of processes, one of which is very important is acetylation. Okay? Basically what happens is that acetyl groups are added to these tails that you saw, and by doing that we get what's called condensation, right? We get opening, and it's a sort of a back and forth reaction that occurs. There is an enzyme, a family of enzymes known as HDAC. Okay, these are histone deacetylators, And there is a complementary set of enzymes that catalyze reactions, actually catalyze the acetylation, that are called HAT. And these are histone acetylators. Okay. When you deacetylate these tails, that causes these nucleosomes to compress together. Okay, and it gives you sort of a compressed area. And when you acetylate them, it tends to open them up. When I want to read the DNA that's in here, so I want to transcribe the DNA and actually make message out of maybe a gene region, a locus that's in here somewhere, the HAT will come in and it will acetylate it, and that will help open up this DNA so that we can get exposed. When you don't want that, then you deacetylate it, okay? Or in areas, remember, you may think we have about 20-something thousand genes that make up a human system. We don't turn all those on at once. I'll say that over and over again throughout the semester, but we don't turn them all on at once. We don't have 20,000 genes active in the palm of my hand. We have a few thousand. The ones that are turned off, one of the ways you can turn it off is deacetylate the histone areas around it, and that will not allow that particular message area to be read. Okay, this is known as the histone code argument. Basically, that you have these two complementary forms, one that deacetylates and one that acetylates, that work together. Now, just pointing out here, they're actually are deacetylators, or HDAC and HAT, that occur out in the nuclear region and then in the cytoplasmic region. Now, ones in the cytoplasm tend to acetylate proteins and activate and deactivate proteins, but very similar types of proteins are actually found that can cross the nuclear membrane here, the transmembrane, and can work inside of the nucleus itself. So it's just an interesting little thing of how this is going to work. And we're going to see in a moment what this condensation means, but I wanted to go ahead and give you at least one of the models about how we can open up DNA and close it up when the cell needs to do that. Okay, and one of those ways is by this relationship between deacetylators and acetylator proteins. Okay, now where have we gone so far? We've taken DNA and we've coiled it. We made some reduction in its length. We now take DNA and we put it into these beads on a string. We get a definite reduction in length formed out, and this is basically the form that we're going to find much of our DNA in when we look into a cell in G1, okay, in any somatic cell. All right, and that's the way it stays until, for the most part, until we get to replication. And then once we go through replication, we start packing immediately. Right? We got everything copied. We start packing, spend all of our time in G2 packing until we get up into finally either prophase of meiosis or mitosis. Okay? That's going to be in the next lecture. We're going to talk about how we go from this structure here, once it's replicated, to these really tiny condensed proteins, chromosomes, that you're going to see as metaphase chromosomes.